Hey, it's Luke from Certis Oncology Solutions. I'm the lead developer for a new tool that we've created. We call it the CertisOI Assistant. The assistant uses the latest in generative AI to help you with tumor model selection for your next preclinical study. Look, at Certis, we understand that tumor selection can be frustrating. Each of your studies requires a specific set of characteristics. Typically, you go to an online tumor database and you attempt to find relevant candidates. You usually have a set of search and filter tools that are very rigid, and if you're looking for things outside of the predefined filters, or for example, you want to search across multiple characteristics at once, you're out of luck. On top of that, the results are usually just a basic list of characteristics, images, and maybe some tables with genetic information. If you want to do any sort of analysis, you have to download the data, if that's possible, and then recruit a scientist to run the analysis outside of the platform. All in all, these tools can be cumbersome to navigate, and in some cases, they're just unable to capture the specifics of your needs. And this is exactly why we built the CertisOI Assistant. We want you to be able to search across our extensive tumor bank with complete flexibility, free from predefined filters and search tools. And not only that, we want you to be able to analyze the tumor characteristics and the genomic datasets without having to leave the platform. The assistant is designed to work with you as a partner, and it's designed to make tumor selection easy and hopefully exciting. So how does it work? Well, you interact with the assistant in natural language. You ask questions and then follow up on the answers that it provides. The assistant will digest your questions, pull the relevant information from our data sets, and unlike a traditional tumor database, the assistant will write and execute code to analyze the information that it pulls. It'll provide the answers in natural language, and it can also provide files for you to download and share, and data visualizations. The assistant is also deeply connected to our Certis AI platform. It has access to over 3.5 million monotherapy predictions, 1.9 billion combination therapy predictions for FDA-approved drugs against models in our tumor bank, as well as cell lines in the Cancer Cell Line Encyclopedia. You can learn more about Certis AI and our predictive models on our website at certisoncology.com. So let me take you through some examples that highlight the capabilities of this new tool. As we run through these examples, take note of how you might complete a similar search and analysis using the tumor databases that you're already familiar with. I think you'll quickly realize that the assistant presents a much more streamlined and powerful experience, and what might have taken you hours on those other systems should now only take you a couple of minutes. Okay, for this first example, let's explore model selection for a preclinical study. In this scenario, we're looking to test a novel compound on colorectal models, specifically models that have a mutation in the KRAS and the EGFR gene. With the assistant, we can just ask for what we want, and it will tell us if there are any models that fit our criteria. So you can see here I'm asking to identify models with co-mutations in EGFR and KRAS. And you just hit enter. Okay, we see that the assistant has found a number of colorectal models with the mutational profile that we're after. But these are just the model names and the model type. I actually want to know specifically what were the mutations that were found. And I'd also like to explore some of the growth kinetics for these models. So let's go ahead and ask the assistant to provide the mutation details and also provide growth characteristics. Okay, so the assistant has provided two tables for us. The first are the mutational details that we asked for. So we've got the specific mutation here, and we can compare amongst all of the models that we've been investigating. Below that, we have the growth characteristics. This gives us a sense of how quickly these tumors might grow in these particular models. This type of exploration is a good example of how you can drill down into the specifics when you're doing model selection. So you can choose whatever characteristics you want and ask the assistant to compare the models, and it'll provide this really clear format for you to compare and choose the most relevant model for your study. So let's take this scenario one step further and talk about therapeutic response predictions. I mentioned before that we have a pre-built library of FDA-approved drug predictions against all of these models. So in this scenario, let's say that we're investigating EGFR inhibitor resistance. So we want to know which of these models that we've so far identified show resistance to common EGFR inhibitors. So you can see I'm going to ask the model I'm going to ask which of these models show resistance to known EGFR inhibitors, and I'm going to tell it to provide both a table and a visualization of the results. Okay, this is really cool. So the assistant has identified 4 different EGFR inhibitors and pulled the therapeutic response predictions against those inhibitors for the models in question. So you can see here it's created this bar chart for us. On the x-axis we have each model, and then on the y-axis you have the therapeutic response prediction. A more negative score indicates higher levels of resistance. So we can clearly see that CRT 287 and CRT 476 show resistance across all 4 of these EGFR inhibitors. And then below the graph, you just get a breakdown of the individual data points that were used to create it. So let me show you one additional thing. The assistant can also pull up histology imaging if we have it. So I'm going to ask it to pull histology imaging for. Now, when I want to specify a particular model, the easiest way to do it is to use the @ symbol. That pulls up a little search box, and then I can just type 287, and there we go. We have the model that we're interested in, and hit @ again and type in 476, and there's the second model. So now I'm specifying that I want histology imaging for 287 and 476. So just hit enter. The assistant pulled up the imaging for these 2 models. So we can see some beautiful Ki67 staining images here, and also some H&E staining. So at this point, we are well on our way to choosing models for our next preclinical study. For the next example, let's switch to another model catalog. You change the catalog down here with this button. We can see that we're currently on the Certis Oncology catalog, but we can change that to the Cancer Cell Line Encyclopedia, or CCLE catalog. Let's go ahead and do that. And for this example, we're going to investigate repurposing of a drug. The drug we've chosen is disulfiram. It is currently approved for alcohol abuse, but the literature has shown that it has anti-cancer properties, and we're going to focus on GBMs. So first, let's ask the assistant what GBM models are sensitive to the drug disulfiram. We can see that there are a number of models predicted to be sensitive to this drug, so we might be onto something. I've also read that this drug leads to oxidative stress in cancer cells that can lead to apoptosis. I want to know whether there's any correlation between expression levels of the genes that are involved in oxidative stress response and the predicted therapeutic response. So a lot of these genes are targets of the transcription factor NRF2. And rather than explicitly define the genes, which I actually don't know off the top of my head, I'll let the assistant figure it out for me. So here's what we're going to ask. Analyze the sensitivity of these top models with expression of NRF2 target genes that are involved in oxidative stress. I want it to create a scatter plot for each gene with a trend line and a correlation coefficient. The assistant gives us a detailed look into these two genes that I've highlighted, what their role is in oxidative stress, and why our therapeutic response correlation may be occurring. So this is a really nice high-level breakdown that can help you understand some of the insights that are surfaced by the assistant. I hope the examples that we covered today give you a sense of just how powerful a tool CertisOI Assistant can be during your model selection. Soon we'll be adding additional data sets that the assistant has access to and new functionality around custom drug response predictions. Thank you all for watching, and I'll see you next time.